I will be talking about the Short Healthy Eating Index. One eating event or single food rarely has a direct impact on health outcomes. Instead, the impact of diet on health outcomes is from complex eating patterns over time, and this is what we mean when we talk about dietary quality. We know that when a person has low diet quality, such as when they consume excessive amounts of sugar or do not meet guidelines for fruit and vegetables, they have an increased risk of developing obesity and other chronic diseases, such as type 2 diabetes, cardiovascular disease, and some types of cancer. Conversely, we also know that when a person has a high diet quality, that is associated with a reduced risk of developing chronic diseases. Because of these associations, dietary quality is often a target in nutrition intervention and assessments. Although there are a number of different tools available to measure constructs of diet quality, the Healthy Eating Index, or HEI, is one of the better known approaches. The HEI score reflects how well an individual's diet compares to the dietary guidelines for Americans. The possible scores for the HEI range between 0 and 100, with higher scores indicating a higher diet quality. The scoring system for the HCRC includes 9 dietary components that Americans are encouraged to eat more of, and 4 they are encouraged to limit. There are no tools that can be administered to individuals, scored, and then used to produce an HCI score before we developed this tool. Instead, first, food consumption had to be assessed using strategies such as food records, 24-hour recalls, or food frequency questionnaires. This step had a high respondent and researcher burden. Currently, all of the dietary assessment methodologies that we have today come with major limitations, but until new, better objective assessment methodology becomes available and accessible, assessment tools that require less respondent and researcher burden are needed. So we set out to develop and validate a short tool that had a lower respondent and researcher burden than existing tools and methods. That could be used to assess diet quality in individual foods consumed. Specifically, we wanted to determine if a Short Healthy Eating Index, or SHEI, could accurately estimate dietary intake of food groups and the HEI score. And we would determine this by comparing the short HEI survey to the Dietary Screener Questionnaire, DSQ, 24-hour recalls derived HEI, and carotenoid measurements. The survey development and validation process had a 3-step process. In the first step, the survey questions were developed and refined. In the second step, a concurrent criterion validation process was used to determine if the diet quality score that we calculated in the short HEI correlated with the 3 existing validated measurements we were using: the HEI calculated from repeated 24-hour recalls, the DSQ, and the dermal carotenoid levels. In the third step, we conducted a confirmatory analysis with a larger sample to confirm that the correlation of short HEI and DSQ that we had previously found. In that first step of developing and validating the questions, survey questions were designed to mirror the components of the HEI scoring system by 15 content experts who worked together to develop the questions through repeated individual cognitive interviews and focus groups. We then conducted cognitive interviews for face validation using a response process approach with a convenient sample of 10 college students, 5 males and 5 females. In the second step, a concurrent criterion validation process was used to determine if the short HEI, which was 35 items at the time, accurately identified an individual's overall dietary quality and also intakes of specific food groups. This was done by comparing the intake estimates obtained from 50 college students by using 3 existing validated measurements: the HEI calculated from repeated 24-hour recalls, the DSQ, and the dermal carotenoid levels. 2 approaches were used to score the short HEI. First, a content expert developed system, and the second being a data-driven approach. Content experts transformed the HEI minimum consumption cutoff scores to meet a 2,000-calorie diet and to be comparable to the servings reported by the short HEI. Because the HEI sets a minimum amount of foods consumed in each group before full points are allocated in the HEI scoring system, whereas in the short HEI, it assessed numbers of servings per day consumed. The content experts adjusted the short HEI servings per day options to match what would be needed to meet criteria to receive full points in the HEI. This means that the short HEI serving responses options were changed into amounts that matched in DSR units in the HEI calculations, such as cups, ounces, teaspoons, or gram equivalents. The content experts then identified calorie consumptions for each of the possible short HEI serving size food groups. For example, 25 calories were identified as coming from half of a cup of cooked non-starchy vegetables, or 80 calories coming from half a cup of cooked pasta. The content experts also identified the minimum amounts required to be consumed from each of the short HCI food groups that would be needed for it to receive full points toward the total dietary score in the short HCI scoring system. All other intake options below that amount were reduced in stepwise approach. The scoring system was then reviewed for practical and clinical accuracy by the content experts. The data-driven approach used a classification and regression tree (CRT) algorithm to extract rules for estimating the intake of each food group and total energy consumed. For each food group and the total dietary quality score variable, the true estimates from the NDSR were used as the dependent variable or target variable, and the short HCI variables as independent variables or the input variables. The CRT algorithm automatically selected short HCI variables to split participants into groups and to determine average consumption for each group. To allow for better generalizability, a minimum size of 20 was required for a node to consider splitting. The maximum depth of the tree was set to 5. Since the dependent variable was continuous, the food group intake, the CRT algorithm provided a path with automatically splitting responses into subnodes, seeking to reduce variance. The CRT algorithm was run using the recursive partitioning and regression tree (RPART) package in R. Data-driven scoring process was then compared to the content expert scoring system to determine if the scores generated were clinically equivalent. After they were determined to be acceptable, the scores for the 13 HCI food components were added, and this is what produced the total short HCI score. Pearson's correlation coefficients were calculated to examine the strength of the short HCI estimates in the 24-hour recalls, the DSQ, and the dermal carotenoid level. In the third and final step, confirmatory analysis was conducted with 398 college students to confirm that the short HCI scoring system produced an outcome equivalent to the DSQ. Balancing the correlation performance and interpretability, the final scoring rules were selected from different possible approaches. Specifically for total fruits, whole fruits, greens and beans, and added sugars, we adopted the expert-developed scoring rules. For total vegetables, whole grains, dairy, and seafood, and plant proteins, we adopted rules derived with the CRT algorithm, using selected relevant short HCI items as predictors, and the remaining ones derived with the CRT algorithm, using all the short HCI items as the predictors. The last group, using a purely data-driven approach, was due to a greatly enhanced performance, whereas seemingly unrelated items were involved in the scoring rules. For example, green vegetables, seafood, nuts, and seeds were used to estimate refined grains. Sodium used fruit, grains, and water in its scoring rules. Saturated fat used sugar-sweetened beverages, grains, and nuts and seeds in its scoring rules. In the third and final step, confirmatory analysis was conducted with 398 college students to confirm that the short HCI scoring system produced an outcome equivalent to the DSQ. Since the DSQ measured a different set of food groups than the HCI or short HCI, to use the DSQ in confirmatory analysis with the short HCI, we trained the CRT models using DSQ food group intake as a dependent variable and using the short HCI questions as the predictors. Pearson's correlation was conducted to look at the relationship between the DSQ items in the short HCI and the DSQ instrument from the original training data, and we found that the food group intake amounts estimated using the short HCI had a moderate to high correlation with the DSQ. We also looked to see if there was a correlation between skin carotenoid measurements and the short HCI scores. We found that the short HCI's estimate green vegetables intake and dermal carotenoid levels, and the short HCI's total healthy eating index and dermal carotenoid levels both had a moderate correlation. So overall, the correlations between the short HCI estimates and the DSQ estimates of the confirmatory sample showed a moderate to strong correlation for all items. We ended up with a 22-item tool because 7 of the original 35 questions that were developed for the short HCI that asked about amounts that were consumed less frequently than 1 time a day were removed during analysis because they were found not to contribute to the overall short HCI score. Another 2 questions were also removed, one about oil and another about salt, because they did not contribute to the short HCI score or to any food group estimation. 4 questions were also removed about meat and red-orange vegetables because they also did not contribute to the short HCI score or any food group estimates. The final scoring system for the short HCI dietary quality score was generated using 17 of the short HCI questions and 1 question on gender for a total of 18 items. 4 questions were then retained in the short HCI because they were found reliable for estimating intakes of specific food groups. Thus, the overall short HCI tool ended up being a 22-item tool that could be used to calculate a short HCI score and estimate individual food component consumption. This 22-item tool is free to use, has low respondent and researcher burden, and can be used to accurately estimate overall dietary quality and some individual nutrient and food group consumption: fruits, vegetables, dairy, added sugar, sugar from sugar-sweetened beverages, and calcium. The accuracy of this tool in estimating intakes of these specific food components may make this tool especially useful in research and health promotion program evaluation, as these are areas of intake associated with disease prevention. Additional validation studies are currently underway to validate the short HCI assessment tool in a variety of populations. Thank you, and here are my references.